In this video, we're going to talk about ribosomes, which are the machines that read mRNA and make proteins. And we're going to talk about the endomembrane system, which is a group of organelles that share the common feature of being able to exchange pieces of their membrane back and forth with each other. So in this image, it's a very simple cell. You see the nucleus. Inside the nucleus is the DNA, and many genes in DNA code for proteins. So what your cell will do is it will copy a gene in your DNA, and when it copies DNA, it does it by making RNA. And so that RNA copy of the gene is going to leave the nucleus through one of those nuclear pores, and a ribosome, which is a complex of proteins and another kind of RNA called rRNA, the ribosome is going to read the instructions in that mRNA, and it's going to make a protein based on those instructions. So the yellow-orange structure that's attached to the red mRNA is the ribosome, and those small orange circles that are coming out of it in a string, those are the amino acids that it's putting together to make a new protein based on the instructions that it's reading. So again, ribosomes build proteins based on instructions in mRNA. So they kind of travel along that piece of RNA and they build a protein as they go. We're going to talk a lot about how proteins are synthesized later on in the course. So right now I just want you to have a general concept of what a ribosome is. So when I tell you there are ribosomes on the endoplasmic reticulum, that means something to you. And ribosomes are found in kind of two general places. They're either found attached to a membrane or not attached to a membrane. So some ribosomes are attached to an organelle that we're going to talk about in a second called the endoplasmic reticulum, or they can be attached to the membrane on the outside of the nucleus, the nuclear envelope. And then other ribosomes are freely floating around in the cytoplasm. Ribosomes have two subunits, a big subunit and a small subunit. I think they look like a hamburger bun. I didn't make this picture, but I appreciate it. Okay, so that's ribosomes. Now let's talk about the endomembrane system. So the endomembrane system is a bunch of membrane-bound organelles. Some of them are big, some of them are little, some of them are directly connected to each other, some of them are separate, but the main thing that they all have in common is this idea that pieces of membrane from one organelle in this system can kind of merge with other organelles and become part of their membrane. So basically all the components, this will make more sense as we go, all the components of the endomembrane system are connected by membrane transfer through vesicles. So what is a vesicle? A vesicle is a little tiny bubble of membrane that can have stuff in it, and vesicles are able to merge with the different membranes in the cell. So maybe the cell membrane on the outside of the cell, for example, a neuron, if those were neurotransmitters inside that vesicle, then that vesicle could merge with the cell membrane in the neuron and it would release those chemical signals to communicate with the next neuron in line. And vesicles can be formed by basically the opposite series of events. So sometimes a little piece of membrane will pinch off around maybe some content, say something that the cell wants to ingest. So it'll pinch off and create a vesicle that contains those contents. So the cell membrane can do this, but also different organelle membranes can do this. So that vesicle can then travel to another organelle and merge with that organelle, and basically we just end up with these little components of membranes that that can travel from one organelle to another. So part of the endomembrane system is the endoplasmic reticulum, and there are two parts to the endoplasmic reticulum. There is the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum is called rough because it's covered in ribosomes. So when you look at it under a microscope, there's lots of little dark dots on it that make it look kind of rough. And remember what those ribosomes do? They make proteins. So the rough endoplasmic reticulum, one of its jobs is to be a place where a lot of proteins are made, and then those proteins can be transported to other parts of the cell when the membrane of the rough endoplasmic reticulum pinches off and forms a little vesicle, and then it travels somewhere else in the cell. The smooth endoplasmic reticulum is called smooth because it is not covered in ribosomes, and it is involved in things like detoxification, for example. We'll get to a slide on that in a second. Also, in this picture, I want you to notice that the rough endoplasmic reticulum is continuous with the nuclear envelope. So the nuclear envelope is kind of a distinct thing, but it also, goes outward and then joins with the rough endoplasmic reticulum. So these membranes are all interconnected. This is kind of a recent research finding on the endoplasmic reticulum, but I thought it was cool. So the endoplasmic reticulum is made up of a lot of stacks, skinny, long stacks of sheets of membrane. But if you were able to look at it three-dimensionally, you would see that it's got a little bit of a structure of a spiral parking garage. So those membranes are all continuous with each other, but they make these long, flat stacks. And I think this pretty much says what we already said, except that if you were to, measure all of the membrane in the cell, all the organelle membrane and this plasma membrane and everything, the endoplasmic reticulum uses up about half of that membrane. So it's a really major component of most cells. And again, this is a microscope view, so you can see the little dark dots are the ribosomes. So they're on the rough ER. Also notice that the rough ER is a lot of flat stacks, whereas the smooth ER makes more like little skinny tubules. And so the rough ER with the ribosomes makes proteins, and that includes glycoproteins, which just means proteins with like sugar chains attached. And again, it can make vesicles that transport those proteins to other parts of the cell, and membrane, if sometimes the cell needs to grow, so the plasma membrane needs more material. Some of that membrane material, those phospholipids and stuff, will be created in the rough ER and then transported to the plasma membrane through those vesicles. The smooth endoplasmic reticulum has a number of jobs. It helps create lipids. It helps break down sugars, or maybe build sugars, actually, I'm not sure. Metabolize can go either way. It is involved in detoxification of drugs and poisons. So I think one of your mastering biology questions has to do with the liver, and the liver detoxifies alcohol. So what would that cell have a lot of? It would have a lot of smooth endoplasmic reticulum because that's doing the job of cleaning out the alcohol. It can be used to store calcium ions, and I'll show you an example of that in just a second. And some, like I said, some cells have a lot of smooth ER if they have a job like they need to create a lot of fat, or they need to detoxify things. So you might see, I can't remember how much we get into this in this class, but sometimes the endoplasmic reticulum has a specialized function and a fancy name. So in muscles, the endoplasmic reticulum is called the sarcoplasmic reticulum. A lot of muscle words start with sarco, like the plasma membrane is called the sarcolemma. Basically the endoplasmic reticulum in the muscle cell stores calcium, and it's part of the system that enables your muscles to contract. So don't worry about knowing that right now, just that's like forewarning for the future, you might see that. Okay, so we said the endoplasmic reticulum makes stuff and ships it off to other places. The Golgi apparatus is another organelle, membrane-bound organelle, that I like to think of as like the FedEx of the cell. So it's kind of it receives packages and then it figures out where they're supposed to go and then it sends them off to that place. So the Golgi apparatus, I'll show you a picture in a second, but what it does is it might take some of those proteins and things that are made in the Golgi and it might tweak them a little bit. It might modify them to get them just right. The Golgi can also manufacture some kinds of molecules, but like the thing that it's known for is sending things off to where they need to go. So the Golgi apparatus kind of looks a little bit like the endoplasmic reticulum, which I think can confuse people a little bit. It also has these flat stacks of membranes. But what basically happens is, say, like a vesicle from the endoplasmic reticulum runs into the Golgi apparatus. The membrane of the vesicle merges with the membrane of the Golgi apparatus. Whatever was inside the vesicle is like dumped into the Golgi, and then maybe it gets modified, and then a new vesicle pinches off with whatever proteins or whatever it needs inside, and then that vesicle travels to where the protein or whatever content ultimately wants to go. So I'll show you another picture. So again, here we've got the nuclear envelope in purple, the rough ER in blue, and you can see that the rough ER is sending a vesicle full of proteins to the Golgi. The Golgi's, I don't know, modifying those proteins some way, chopping them up or adding something to them or whatever. And then the Golgi sends out a new vesicle, and that vesicle can either, say, merge with the plasma membrane if that's what we want to do, or it can go to another part of the cell. So yeah, so the Golgi is the shipping and receiving and sorting and just getting everything to where you want it to go. So the next couple of organelles that we're going to talk about, when I was looking online, it seems a little more questionable whether people consider these part of the endomembrane system or not. So some people do, some people don't, but we'll get into them. So lysosomes are, they're kind of like large vesicles that are full of digestive enzymes. So you can kind of think of them as being analogous to the stomach in your cell. When something in your cell needs to be digested because, say, it's broken down and it's no longer working, or you just don't need it anymore, then it goes into the lysosome, and the lysosome digests it. One thing lysosomes might do is digest food that's eaten by phagocytosis. We'll talk about this more later on in the class, but first I just want to show you the picture that goes with this. So say the cell ingests a little piece of food by pinching in and forming a vesicle around that piece of food. Then the lysosome, which has digestive enzymes in it, can merge with that vessel or vacuole. We'll talk about vacuoles in a second, but a vacuole is like a big vesicle. The digestive enzymes can merge and break down the food, and then the cell can use that broken down food for whatever it needs to do with it. Also, lysosomes can be involved in breaking down parts of the cell that no longer work. So here we have a broken down mitochondrion and a broken down peroxisome, which is another organelle. And so the lysosome merges with that vesicle, and it digests the parts of the mitochondria and the parts of the peroxisome, so that the cell can either like get rid of them, like kind of spit them back out, or can use the parts to make new mitochondria or peroxisomes or whatever. So what is a peroxisome? A peroxisome has some similarities to a lysosome. It's a compartment that produces hydrogen peroxide. So that's why it's called a peroxisome. And that hydrogen peroxide can be used to help break things down. It's kind of another digestive compartment in a way, but it has a lot of different functions, and it seems like one of the components that still isn't super well understood. So these are just some of the jobs that a research lab that studies them says they can do. You don't have to know all these, but the main idea is that like peroxisomes have hydrogen peroxide. They can help break stuff down. They do a lot of other things. We're still figuring out exactly what all they do. And some people say peroxisomes are part of the endomembrane system. Some people say they're not. It seems like people are moving toward, yes, they are. But anyway, vacuoles. Vacuoles are large vesicles derived from the endoplasmic reticulum and the Golgi apparatus. or I assume like one or the other. The solution inside a vacuole is different from the cytosol. So again, that membrane around the vacuole separates the inside from the outside so that the inside can be a little different. And there are several different categories of vacuoles. So a food vacuole is kind of like a large vesicle, a large membrane bubble that's wrapped around food that the cell ate through a process that we call phagocytosis, which is like cells taking in particles, large particles, solid particles. Contractile vacuoles, I will show you. This is kind of cool. This is, so the contractile vacuole is the sort of sun-shaped region in the upper left third of this. I think this is a paramecium. So contractile vacuoles fill up with water, and then the cell can like contract them and squeeze out the water. So it helps a cell, certain kinds of cells, not all cells have these, but it helps cells that have them control how much water they have inside of their membranes. And then central vacuoles are big components of plant cells. So central vacuoles also can fill up with water, and they help make sure that the cell is like rigid. So when you don't water your plant, what happens is that central vacuole, all the water in it gets used up and it starts to get smaller, and so your cell doesn't have that pressure pushing out on the cell walls that like makes it rigid, and so your plant starts to wilt because it can't stand up strong and tall because it doesn't have that water pressure holding it up. Then when you water your plant, that central vacuole fills up again and that pressure gets exerted on the walls of the cell, and so the plant can stand up straight and tall. And yeah, so that's vacuoles. So the endomembrane system is a little bit confusing because sometimes the lines between some of these features can be a little bit blurry. So the Golgi apparatus is always the Golgi apparatus. The endoplasmic reticulum is always the endoplasmic reticulum, either smooth or rough. But the vesicle versus vacuole, sometimes even versus lysosome. I don't know about peroxisomes, but because they're always like merging with each other and stuff, it gets a little gray. So understand those basic categories of, you know, what's a vacuole, what's a vesicle, what's a peroxisome, what's a lysosome. But just know that like when you see pictures of them in the diagram, some of them might be a little bit hard. They might all look like each other. Like they might just look like bags of membrane, and there's a reason for that, because the cell is a fluid dynamic system, so things can be in flux. Anyway, I hope that made sense. There you go.